Hello everyone. Today, I'd like to share our recent work on an oral vaccine for allergic rhinitis that harnesses the gut-nose immune axis to provide durable protection against allergic airway diseases. Allergic rhinitis (AR) affects over 400 million people worldwide, and its prevalence continues to rise with urbanization, dietary changes, and gut microbiota dysbiosis. Although current therapies, including antihistamines, intranasal corticosteroids, and sublingual immunotherapy, SLIT, relieve symptoms, they rarely induce durable antigen-specific immune tolerance. Moreover, SLIT is limited by prolonged treatment and poor adherence. Therefore, new strategies to establish long-lasting immune tolerance are urgently needed. To explore the link between dietary fiber and AR, we analyzed 8,092 NANS participants. Higher fiber intake was associated with a lower risk of AR, with each 10g/day increase reducing AR risk by 13.4%. Mechanistically, dietary fiber is fermented by the gut microbiota into short-chain fatty acids, SCFAs, which promote regulatory T cells and regulatory B cells expansion through FFAR2 signaling to restore immune tolerance. Because direct SCFA supplementation has limited efficacy, we hypothesized that combining dietary fiber-mediated SCFA production with targeted allergen delivery could provide a more effective strategy for inducing long-lasting mucosal immune tolerance. Based on these findings, we developed a nano-dietary fiber platform, NDF, that combines antigen-specific immunotherapy with sustained gut SCFA production to induce durable oral immune tolerance. Fabricated from beta-glucan, NDF serves as both an allergen carrier and a fermentable substrate for the gut microbiota. The nanoparticles exhibited a uniform spherical morphology, 89 nanometer, an encapsulation efficiency above 96%, and prolonged intestinal retention of the allergen. Mechanistically, NDF activated regulatory immune pathways, while butyrate inhibited dendritic cell activation and enhanced IL-10 production, establishing a tolerogenic intestinal microenvironment. We next evaluated NDF in an OVA-induced mouse model of allergic rhinitis. Oral NDF significantly reduced sneezing, nasal OVA-specific IgE, and allergen-induced nasal inflammation. It also increased IL-10 while suppressing Th2 cytokines, including IL-4, IL-5, and IL-13, and markedly alleviated eosinophilic infiltration and tissue damage. Mechanistically, RNA-seq revealed activation of immune tolerance-related pathways, while mass cytometry showed that NDF restored intestinal immune homeostasis by reversing the imbalance of immune cell populations. Together, these findings demonstrate that oral NDF remodels the intestinal immune microenvironment and effectively protects against allergic airway inflammation. We next investigated whether NDF-induced regulatory immune cells migrate from the gut to the nasal mucosa. Using KICGR photoconvertible mice, we found that NDF markedly promoted gut-to-nose immune cell migration. RNA-seq revealed a tolerogenic phenotype in these migrated cells, and 73.4% of IL-10 producing cells were CD19 B cells, identifying regulatory B cells as the major mediators of gut-to-nose immune tolerance. In FAR2 deficient mice, NDF completely lost its immunoregulatory effects, demonstrating the essential role of the SCFA-FFAR2 pathway. Notably, an 8-day oral treatment provided protection for at least 4 months, indicating durable immune tolerance. Our study presents 2 key advances. We developed an oral tolerogenic vaccine that co-delivers allergens and dietary fiber to induce durable antigen-specific immune tolerance. We further identified gut-derived IL-10 producing regulatory B cells as the major effector cells that migrate to the nasal mucosa through the gut-nose axis. These findings provide a promising strategy for the long-term treatment of allergic airway diseases.